Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Hello viewers, welcome to the TV learning series, and here we are again, Senior Sixes. We are looking at the process of natural selection and artificial selection like we've been looking at in the previous lessons, but today, as we come towards the end of the unit, 1 or 2 lessons to go, we are looking at how artificial selection can relate to evolution, as we're yet to see, and how a change in the natural environment by artificial means. In this case, we are looking at the use of antibiotics, industrial pollution, resulting into a new environment determining who survives and who does not based on the changes they have acquired because of that change in the environment. So, this is where we look at industrial melanism and antibiotic use. We've used antibiotics, I don't know for how many ages in life. Since the First World War, antibiotics have been in action. Treatment of wounds in war victims and war incidents, accidents, injuries, all the way through bacterial infections. But the question is, how many times have these bacterial forms appeared? And if they do appear, are they showing any resistance to the medicines? What about the Industrial Revolution? Polluting environments, what is the effect on insects, and how have they come up with differences? And with these differences, the question is, how has the nature actually determined at that point who survives and who does not? So, let's start off with antibiotics and bacteria. We cough most of the time. We have bacterial infections. We have tuberculosis in action. We have very many bacterial diseases much around us. A reason being, possibly, these bacterial forms keep changing all the time. The question is, who is accelerating the change? Who is favoring the transitions from one form of bacteria to the other? So let's look at one. In all forms of selection, reproduction is key. And once a population has resulted, there may be differences. These variations are passed on again in reproduction. But the question is, what is the role of antibiotics in this? So we do have our bacteria producing, and we now look at new forms having new shapes. As you can check, these are differences. If antibiotics are able to lock onto certain forms of bacteria, will they be in position to lock on others? If this is able to treat a bacterial infection of this type, strain of bacteria, and all this because of the nature of the components it has as being receptive to the sites on the surfaces, the antigens on the bacteria. Now the question is, who survives and who does not? If the antibiotic in this shape is able to be working effectively and clears out these 2 strains of bacteria, so it means that this strain and this strain as being different from this one, that have all come from this, means that here, this antibiotic with this shape is effective. Because the bacteria are unable to resist it. So it clears them out of the population. So they are killed. The question is, what about this other variant? Has a chance to continue reproducing and can continue multiplying? We therefore categorize this as a resistant strain, which survives the application of antibiotic. Now, the question is, if the application has cleared this and has not cleared this, what is going to happen next? So there we are. The resistant bacteria cannot be killed by the antibiotic. So it gets a chance to produce and produce and produce and increase its level of frequency of the strains it has. So the population grows, meaning that treatment of a bacterial infection of this strain with this same antibiotic is not going to work. So if a resistant population grows, we are now looking at one, is this antibiotic, is this medicine that is available as the only option for treatment of bacterial diseases working? If it was working before here, and these two that come out are able to be killed, what about this new variant that has come up? How far is the pharmaceutical industries going to be able to find the remedy, to be able to find another cocktail? That if this antibiotic has two mechanisms which it is able to deal with two forms of strain, look at the shape. This and a V, meaning that the same antibiotic can deal with two forms of bacterial infections. That's what you need to look at. Now, the thing is, what about the third? Are we able to work with this same antibiotic? No. Reason? Look at the receptor sites. Look at the antigens. So there is no compatibility in terms of receptor site, in terms of the shape of the molecules of the antibiotics to be able to lock heads with the surfaces of the bacteria, render them useless, break down their cell walls, weaken them. But we also need to remember that these chemicals, one, puncture the cell walls, two, distort the DNA of the bacteria, and three, change protein synthesis mechanism. So if new proteins to form part of the cell wall of the bacterium breaks down, then so much water enters into the bacterium, and this will reduce the level of rigidity of the cell wall of the bacterium, and so maybe that's the reason as why they say when you're on antibiotics as antibacterials, then you need to take lots of water. Purpose is, can you increase water intake by the bacterial cells, whose cell walls have already been weakened, and then they, boom, burst. So here the population is increasing. So we are now having a new strain compared to the first one because of the differences we have, unable to be worked on by the antibiotic. 2 out of 3 are cleared, 1 stays, increases in population. Now it means that when we do have failure of antibiotics to deal with the strains of bacteria we now have, whoop, evolution is taking place now. A change in form, structure, nature of our organism over a period of time. Boom, there it comes. Now we need to start thinking, what exactly is happening? When resistant genes proliferate, when they elongate, when they duplicate and produce many copies of that, they become dominant in the population, in a community. So they are stretching, they are outbursts, and their levels of infection, if they are dangerous, then it means we are very much exposed, vulnerable, and we do not have an antibiotic to work on it. That's what we are looking at. Now we need to start looking at how many times people have changed antibiotic use over time. Amoxicillin, tetracycline capsules, penicillin, all those forms of antibiotics. The question is, do they still work? How often do you take umuravumba? A base, able to change the pH under which the bacteria do survive in your throat. So when you take it, that's actually a natural antibiotic. Do you have that shrub in the backyard? How often do you run for use of antibiotics in pharmacies? A slight cough, oh, antibiotic, antibiotic, antibiotic. The question is, the more you take them, what are you exposing yourself to? Are you giving a chance these bacterial strains to have exposure to the antibiotic? And possibly, based on the way you take these antibiotics, poorly, without medical consultation. So sometimes you even don't know which antibiotic you're taking for which bacterial infection. What about the farmers? How much of the antibiotics are they using in agriculture, either crop or animal husbandry? So we find that these plants, these animals are exposed to antibiotics for a very long time as they treat disease. That's one observation. Secondly, when we take animal products, when we take crop produce, are they having some of these antibiotics already with them? So when we take the food and respiration goes on and metabolism goes on, are these antibiotics metabolized? Are our cells exposed to these antibiotics? 3, the exposure of these antibiotics all the way through. At one point when we get a bacterial infection, could this be the same related strain that has been exposed to these antibiotics long before, that by the time we actually have that very infection, these same bacterial strains already developed a resistance, already worked out a mechanism to produce enzymes able to break down the antibiotics, that when we finally take them for treatment, they actually are considered not productive. So we talk of artificial selection, we talk of change in species, we talk of speciation, we talk of new varieties of organisms coming up. The question is, how much hand are we putting into that for that to happen? So even when we are looking at antibiotic resistance as a topic, evolution, artificial selection, the take-home point has to be, what is your role in that? How often do you take medication that is an antibiotic? 2, is it a prescription from the medical worker? 3, are you taking it in the very expected way? Do you finish the dose? Because if you don't and some survive, are you going to use the same antibiotic the next time you fall sick? Or the few that survive will have worked out a mechanism to break down the antibiotic as well. That's your homework. All right. The other part we are looking at is, okay, industrial mechanism. Something like that. Moths, butterflies. How many different forms do you know? Which now takes us to, again, differences as a result of pollution. Again, human activity. Agricultural revolution, industrial revolution. So the story goes like this, that long time ago, when people were transiting from agricultural practices, domestic ones, into industry, there was so much demand for energy. So what happens when we talk of the advancement in civilization, from hunting, fruit gathering, to farming, and then now industry? The Great Britain you used to hear about before partitioning and honor remain with Britain, we now call England. So much industrial revolution taking place, much of the energy source is basically coal, and so they dig, they extract the coal and burn it. The steam engine, choo 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 choo. Industries, smoke, soot all over the place. Guess what? In forested places. And what happens is that the soot coming from burning of coal in all the industries that were being stored all over the place end up producing smoke, soot, dark, black. And what does it do? Pollutes the environment. The whole place becomes acidic. And then the acid rain starts killing the lichen. So it means, therefore, that if we go back to our ecology, pollution indicators, that's what we are now looking at. The number or the distribution of lichen in trees all over. Every time you are at school, try to look around the compound. Check the backs of the trees. If there are any whitish things, fungus combined with algae to form lichens. It's a very perfect, close association, intact. None survives without the other. In the presence of high levels of pollution in the atmosphere, the rain becomes acidic. So every time it rains, the lichens are destroyed. They dissolve. They break down. So the reducing number of lichens in any forest, in any trees, or in any compound around would tell us how polluted the atmosphere is. That's one indicator. So looking at the background, in the picture here, follow the laser, and you should be able to see the plan of where it goes. What do you see? That's a butterfly. That's a moth. Then we have another form here, black, hiding in the background, that is darkened by industrial soot, high levels of pollution, removing the grayish part of the lichen. Now, question is, If a predator was to come between these two, which one would it easily sight? Moving this one here and take this one here. This is the initial natural environment, and this is now our new environment out of Industrial Revolution. Now the question is, with this high level of pollution, the gray-peppered moth were easily traced by the predators. So the woodpeckers, the pecking birds, and all other birds that would depend on the insects would easily do what? Trace it, identify it easily. Because the background is now black, and we still have our gray moths. Guess what happens? They are easily seen. So, now we are having pressure determining the direction of selection. The predation from birds and other animals, as they are resting on the black background, since it is now changing. Now, over a number of generations, the dark Carbonaria form of peppered moth has increased with frequency, increasing industrial activities, increasing levels of pollution. So we are now looking at a change from one moth to another as a result of mutations. That is one point you need to take. That the conditions that were prevailing at the time when industries were mushrooming could have produced environmental changes that triggered a change in the DNA. Change the genetic code, change DNA replication, change translation, change proteins, change structures. Now, these small accumulative changes in the DNA we call mutations could have resulted into formation of a rare one, moth, Carbonaria, as a result of carbon pollution and other substances. So now the effect is that the new forms that have resulted out of that level of pollution and as a result of mutations are now going to be favored. So how is the favor coming? Camouflage from birds against the dark trees and branches. They are given a chance to survive. They grow, reach reproductive age, and produce, pass on their new trait of the black peppered characteristic onto new generations. So if our new environment, the industrially polluted region, is favoring the black peppered moth, it means that the gray ones that are still there that did not mutate are easily found, eaten by birds, and very few would do what? Would survive. So over a number of generations, the Carbonaria form, will now have a chance to produce. So the increase in frequency of the genes, their numbers get bigger because they are favored. While these would possibly have to migrate and go to a place where there is less effect of industry, that they can be able to keep camouflaging against their gray background. So this time here we are having two environments, industrial region, not yet industrial region, and industrialized. So now nature chooses, okay, go here, go there. You don't go, then you're going to be finished. What are you trying to say here? That environmental policies in Europe, the question is, what about in Africa? They are looking at markets, they are looking at so much mushrooming industrial activity. The question is, are there any environmental policies that are protecting some of these species? Can we get back our original organisms. Are the policies favoring economies at the cost of nature? That's what I wanted you to look at when you look at this highlight. What about in Africa? So with that, we wrap this with an activity. So get your camera or phone, take a snapshot. That's one. The second is this. Remember that we are looking at the process of evolution interconnected with artificial and natural selection. So I want you to look at these two activities. Go back, take a snapshot of this, and then we come here as well and be in position to attempt. But my major interest lies here. The purpose for putting all these questions together is that these two components are interconnected. So with that, we come to the end of the session, and thank you so much for following. That is your activity for you to do. I hope you'll be able to find out. Please read more. This is purposely for giving you an overview of what you're supposed to do. Thank you so much.